Hello, I'm Professor Paul Bingham, and this is Biochemistry 1, and our topic today is mitochondria and electron transport. So let's put this into context with running up from what we've learned over the last several sessions. So let's remember some basic terminology first, that the half of metabolism in which complex reduced hydrocarbons are oxidized to liberate energy is referred to as catabolism, and the oxidative phosphorylation and membrane mitochondrial electron transport are central to catabolism. Let's be a little more specific now. So burning different kinds of foods, amino acids, glucose, fatty acids, and others, all ultimately feed into the system that we're going to talk about today. We have focused in the last several segments on the metabolism of glucose itself, where glucose runs through glycolysis and into the TCA cycle. And the TCA cycle in turn is going to run into electron transport and oxidative phosphorylation, our subjects for today and the next segment. But notice that degradation of fatty acids and amino acids, and indeed other molecules not shown here, all flow into the TCA cycle and therefore into electron transport and oxidative phosphorylation. So the process that we're talking about in the segment today and in the next segment is the central hub in which all reduced hydrocarbons, all the different forms of reduced hydrocarbons in the food that we eat come to be catabolized and turned into energy, if that is their fate, rather than being used to assemble macromolecules. What we want to look at now is the next step in this process. That is when the energy in the form that's extracted from the TCA cycle, the citric acid cycle, I'll remind you of what that is in a moment. We talked about it, of course, in earlier segments, is in turn converted ultimately into ATP. Let's get oriented a little bit here. So notice that both glycolysis and the TCA cycle are reducing NAD. They also reduce FAD, a different related redox cofactor, but NAD is the primary one, and so for simplicity we'll focus on that here. We'll come back to FAD and where it appears in a few moments. And then the purpose of oxidative phosphorylation is going to be to take that reduced NAD and oxidize it back to oxidized NAD. So we have a cycle here, but it's not a futile cycle where we're just reducing NAD and oxidizing NAD. We are in fact extracting reducing potential from food, storing it in reduced NAD, and then we're going to use that potential, that reducing potential, to convert it into ATP, a high energy molecule which we can use for all kinds of purposes, some of which you've already seen, muscle contraction, for example. And we're going to do that by electron transport, in which the reducing potential in the form of electrons, high energy electrons from the food we eat, are going to be transferred to molecular water, molecular oxygen, converting that to water. So let's be very clear. The part of this process we're focused on here is going to be the electron transport, which is a subset of this larger process lumped together under the rubric of oxidative phosphorylation. That is where oxidation of our food is used to phosphorylate ultimately ATP. And so we're going to be focused today on the electrons that we extract from food and ultimately donate to water. Donate to oxygen to make water. How are we how are we going to ultimately store that as energy, which can be extracted to synthesize ATP? Okay, so let's zero in a little bit further. So this is an artist's schematic of a cell. Here's the cytosol, the large sort of ocean-like component of the cell, and the mitochondrion. One is indicated here. Let's blow that up. This mitochondrion, as you are probably aware, is an ancient evolutionary descendant of a bacterium that was captured as an endosymbiont in the process of the evolution of eukaryotic cells probably 3 billion years ago or so. And so the mitochondrion is in some sense a bacterium. So many aerobic bacteria do the same, very similar oxidative phosphorylation processes to the ones that mitochondria do. But of course, we're mostly going to focus on an animal mitochondria, including, of course, human mitochondria. So something we won't talk a lot about is that there are transporters that transport pyruvate, remember the end product of the degradation of glucose in the cytosol, as well as some other things, some amino acids, for example, import pyruvate into the mitochondrion and will allow ATP to be to be transported out. So ADP and phosphate are transported in, ATP is transported out, pyruvate is transported in. The pyruvate is going to be oxidized all the way to CO2 and water and will escape as small molecules. Our focus today, though, is going to be on that system for electron transfer. We're not going to worry too much about these transport systems. Just be aware that they exist. Let's focus instead on the structures of the mitochondria that are more directly pertinent to what we're concerned with here. Let's start with the matrix. Notice that the mitochondrion is a double membrane structure. It has an inner and outer membrane. The matrix is the innermost part of the mitochondrion inside both membranes. That is where, for example, the citric acid or TCA cycle occurs. The inner membrane immediately surrounds the matrix, and notice as it's sort of indicated here in stylized form that it's highly convoluted to create a lot of surface area, and all that surface area is going to be it functions predominantly in this process of electron transport that we're going to be focused on today. The more surface area, the more efficiently you can engage in the electron transport processes that we're going to talk about. Okay. The inner membrane space is the space between the inner and outer membranes. That's also going to be very pivotal in the processes that we're going to talk about today. And then the outer membrane surrounds the inner membrane space, inner membrane, and the matrix. So electron transport goes on exclusively in or on the surface of this inner membrane. So this inner membrane is going to be where all of our action is today. We're going to be looking at that membrane and to some extent the matrix and the inner membrane space immediately surrounding it as we look at electron transport. The citric acid cycle goes on in the matrix, as we said, and that's going to provide most of the reducing potential to drive electron transport and the things that ensue from it. Let me introduce you for the first time something you'll see over and over today. As electrons are transported through the electron transport system, I'll sometimes slip and call it the ETC for electron transport complex, ETC for short. It's a commonly used abbreviation. As electrons are pumped through the ETC components in turn are going to pump protons from the matrix out into the inner membrane space. Now that seems like a kind of peculiar thing to do, but as you'll see over the next few minutes, it makes a beautiful sense, and it's a really elegant way to solve the problem of taking reducing potential from food and turning it ultimately into ATP. So the resulting proton gradient that results from using the energy in electrons to pump protons out of the matrix into the inner membrane space is going to be ultimately used to synthesize ATP. That last step of turning that proton gradient into ATP is not our focus today. That'll be the next segment. What we're going to do today is to look at all of the elaborate machinery of electron transport that is used to create that proton gradient in the first place. An extraordinarily useful gradient it is. It's used, in fact, to drive a number of processes. Our focus here today will be its use to drive ATP synthesis. So in the next few minutes, we're going to dive into the details. And this lecture will probably be relatively long, perhaps an hour and a half or so, because there's a lot of detail to be assimilated. I would encourage you to watch it through once, get the big picture, understand as much as you can of the overall context, and then come back and watch individual segments to refresh your memory and reinforce your command of all the specific different details of how this rather elaborate process occurs. It is so elaborate to try to be efficient in capturing as much of the energy in it as stored in a glucose molecule, for example, in the form of reducing potential, captured in the form of high energy electrons, to try to capture as much of that energy as the cell can and as efficiently and completely as it can in order to make the proton gradient, which is in turn going to make ATP. All right. So this is the environment in which we're operating. I'll call that back in a moment. So let me get you in a little bit more in context, a little bit better sense of context. So this is the diagram you've seen in earlier segments of glycolytic degradation in the cytosol of glucose culminating in pyruvate. Then pyruvate jumps into the mitochondrion through one of these transporter systems and then is ultimately degraded to CO2 in the mitochondrial matrix, the citric acid cycle. The question for us here is, if we're going to use reducing potential to make ATP in the mitochondrion, don't we